Okay, so everyone, thank you for coming today, joining the meeting. I'm Rachel, the restoration technician at the Ventura County Resource Conservation District. I'll let my coworker introduce herself real quick.
Hi, everybody. My name is Jaya Bibi. I'm the conservation ecologist here, also at the Resource County Conservation District. Happy you guys can join us.
So we have a few different presentations today from some folks doing some incredible research and projects. And I will turn it over to our first guest speaker, Kyle, and I'll let him introduce himself and kind of talk about his research he's working on.
Okay, hi. Can you all hear me okay?
Yes.
Great. Thank you. So yeah, my name's Kyle Nessen. I'm a monarch biologist. I've been working in monarchs now for about 4 or 5 years. What I'm presenting today is the results from my master's project, which I completed in October, where I looked at how wind affects monarch butterfly behavior at overwintering sites. Okay, let's click here. So just to begin, right? So monarch butterflies go through this amazing annual migration where individuals from all over North America, particularly here in the West, on the west side of the Rocky Mountains, going up into Canada, they will breed in the spring and summer months. And then when fall comes, they will collapse back to these overwintering sites, primarily along the coast. That's what all of these little orange dots here are. These are named overwintering sites maintained by the Xerces Society. There's over 400 as of today. And as you can see, they're all like immediately coastal and they have a few characteristics. And so, the picture I have here on the left is of Pismo Monarch Grove. This is one of the top 10 important sites routinely across California. And it has a lot of typical characteristics. So one, it's a relatively small grove. It's 12 acres, dominated by blue gums that are about 150 feet tall. They create this kind of sheltered, protected area. The butterflies tend to occur right over here, again, immediately coastal. And we think that this pattern happens for particular reasons. In the literature, we call this the microclimate hypothesis, and it states that there are a set of abiotic conditions, things about the environment where these overwintering sites occur that make it quite suitable for monarch butterflies that are, I wish I had a little more time to go into it, but basically they are trying to conserve their energy during the winter months when their primary resource of milkweed has disappeared across the landscape. And so that when spring happens again, they'll have enough energy to go out, reproduce, and start the whole reproduction cycle again. So this hypothesis predicts that these conditions determine habitat suitability. The first one is mild temperatures. So we think butterflies are avoiding freezing temperatures. We know they can't tolerate them for long periods of time. They could dip down into sub-freezing conditions, but no more than a few hours or at the most a few days. But they also are avoiding these excessively warm days because they're ectotherms. Their rate of energy use increases with their body temperature in this nonlinear exponential way. So they're really trying to find the sweet spot between not being so cold that they can't fly or freeze and fall to the ground and die, but not being so warm that they're just like burning through their energy reserves. The second one is high humidity. So monarch butterflies, like all insects, are subject to desiccation. They dry out, they get thirsty. Occurring in a high humid place mitigates that, and being on the coast is a natural place to be where we have this routine coastal fog that comes in and creates not just like more humid conditions, but also surface water in the leaves that they could potentially drink. And we talk about this concept of dappled light in monarch conservation. That's where the structure of the forest, the grove itself, creates this heterogeneous, this like variable light that changes throughout the day. Monarch butterflies are really good at taking advantage of this direct sunlight. They can warm up to favorable body temperatures in a matter of minutes, like low minutes, like single digit, under 5 minutes. And so they like to use this light as a way to behaviorally thermoregulate. And then finally, we historically have thought that wind protection has been like the central thing that we should manage for in groves. It makes sense if you're a small butterfly that weighs like just a few grams, being blown around in these strong 25-mile-per-hour winds could be quite disruptive to you. And so taking shelter in these large forested areas, particularly during the winter when we have our strongest weather storms, weather systems, makes a lot of sense. This idea that wind matters comes from the studies that researchers at Cal Poly did in the late 80s, early 90s, particularly Kingston Leong, who did this like foundational work where he went to groves in Los Osos, like Montaña de Oro, and also by Pismo Monarch Grove. And he went to trees and he measured these like short wind averages, about 10 seconds at the base of the tree where he saw butterflies occupying, and then also nearby trees that he did not observe butterflies on. He compared these and he found statistical differences, but it's like worth mentioning that the highest observed speeds that he had during the study was about 3.5 miles per hour at the ground, and he never directly quantified this behavior. But in that original study, he makes this assertion here that during winter storms, butterflies clustered on those few trees that offer the greatest protection against winds of about 2 meters per second or 5 miles per hour or greater. And over his career and subsequent like studies and observations and experience, he concludes to this 2016 report to CDFW on the management of overwintering groves that winds greater than 5 miles per hour are disruptive to butterflies by blowing them from their roosting branches or dislodging them by shaking their branches. And this idea that wind above 5 miles per hour is incredibly disruptive to butterflies has become really influential in conservation circles, particularly in the management of overwintering groves. Here are a few management guides that lean heavily on this microclimate hypothesis for making recommendations, particularly about wind. This report right here, which I worked on with Althouse and Meade in 2023, we actually went so far as to reconstruct the overwintering grove in Santa Barbara at Elwood in the computer. That's what these like figures are here, and simulate using computational fluid dynamics what a strong storm from the southwest would look like. And that's what these results are. And so what we were doing is we were saying, okay, the grove is functional, maybe marginal based on this threshold. If we add all of these trees that we plan to do and allow them to grow up, how does that change the wind? It makes it much better. And that directly informed the location and density of tree plantings that we did. So this is really, when I say the key thing that we're managing for, I mean it, that we really take wind seriously. The issue with this like microclimate hypothesis, though, is that when other researchers tried to replicate some of the predictions that come from it, they were unable to do so. So Sani and Villablanca, my co-author and advisor on this project, his grad student and him in 2022 tested this directly. So they went to 8 different groves, or a number of different groves, from Morro Bay all the way down to Harbor Boulevard in Ventura, down in your neck of the woods. And what they did was they set up this array of monitoring equipment. So they put a temperature logger, a light logger, humidity logger at the location where the butterflies are within the grove. There we go. And then systematically on the interior and exterior. And what they found was they couldn't actually find anything special between this butterfly spot and these other interior spaces. Only the southeast edge looked different. And that's because it was the one getting the most direct sunlight and getting baked in the sun. So it had more light, higher temperatures. And perhaps more importantly, each one of these groves looked different from each other. So not only could they not find the special spot within the grove that would predict where the butterflies are, each one of these groves looked different, suggesting that there isn't one magic set of numbers or magic set of range of conditions that butterflies are looking for. But critically, they did not investigate wind in their study. And so that's what we set out to do. We're really picking up where that study left off. We're the first study to directly evaluate the disruptive potential of overwintering butterflies of wind. And we really wanted to understand, is this 2 meter per second threshold, this 5 mile per hour number that's widely used in conservation, actually impacting butterflies, or is there more to learn? So the way we did this is we went to Vandenberg Space Force Base, where we had access to a number of different overwintering sites. There's 30 named ones on site, of which like 12 are regularly accessible throughout the year. And we set up these remote monitoring poles. Here is a camera. So I took these trail cameras and I modified them so that they can better contrast between the butterflies and the leaves. It uses near infrared light. It just makes it easier. Like you have a dark spot where the butterflies are against a bright foliage background. And then above that, we have a wind meter that's logging wind conditions every minute. We put them on these tall poles so we can elevate them to the height of the cluster. That's where the butterflies are in this picture. They could be as high as like 40 feet tall. It was really fun to see them in the field. And then here's like an example photo of what they look like. And just to give you a sense of scale, here's my research assistant, Skylar, at the base of my pole with our little ladder to help us set up. This one was about 35 feet tall, and it's all held together with 3 ground anchors that we use, guy lines, to keep it upright. And this equipment, like we would set it up once, we let it run for weeks to up to like a month and a half or so, and it would take a picture every 30 minutes to count butterflies. And we set up a ton of these poles. So here's Vandenberg Space Force Base. These orange dots are all the locations that we set up. So here's one at Minuteman, and here I am setting up at Tangier, I believe. As we go down the line, there's Skylar again at Archery. Some really fun, beautiful places. Primarily, all of our equipment and observations were at Spring Canyon. Like here I am standing next to one of my poles. And I just threw this one in there just to show you that we did lose them along the way. They didn't all quite make it. So I did this over 2 years. There was the 2023 to 2024 season and the 2024 to 2025. I think I said that right, 2023, 2024. Anyway, the first season was not a great year, but it was historically like more of an average. I think there's 300 something thousand in the state. And most of those butterflies on Vandenberg Space Force Base occurred at Spring Canyon, which is this site right here. Unfortunately, the next year, it was the second lowest ever on record, and no butterflies showed up to Vandenberg at all. So even though we had all of these like poles set up, I just have many tens of thousands of images of eucalyptus leaves and wind measurements, but no butterflies, unfortunately. So this is the caveat of the study. All of the results I'm presenting are from one season and primarily from one grove, which is Spring Canyon, which historically has been the most important site on Vandenberg. So once we had all these images, we had to turn them into a number of some kind. It was impractical and infeasible to count them directly. So we came up with this grid system where we wrote custom software and asked undergraduates to help us label these images. Essentially, what we're doing is dividing the image into these grid cells and classifying them by order of magnitude. So this light yellow means there are 1 to 9 butterflies present in there. Orange is 10 to 99 butterflies, and then red is more than 100. And we took a conservative way of counting them. So we didn't take 5, we took 1. We took the lower bound, multiplied it by the total number of cells to get us a thing, added them all up. We had 3 cells over 100, so that gave us 300. So then we had this like pseudo-continuous number of 436. And to measure how the wind conditions or other factors cause the butterflies to change in abundance, we would take 2 samples. So we had a before and after. We have different time windows that I'll explain in a minute. And we would simply subtract them to get a single measure that has the nice property of, if it's negative, that means butterflies went down in size. And if it's positive, they went up. And while we were going through the images, we noticed that direct sunlight seemed to be predictive of these changes. And so we added a second way of counting where we'd add this little yellow border to any cell that had butterflies that was currently experiencing direct sunlight. And so we would have a total, like the total for the image, and then we would separately count how many of those butterflies are receiving direct energy from the sun. So for our analytical approach, we consider two temporal windows. We looked at the finest resolution that we could, which is this 30-minute window from one photo to the next. What happened in that time frame where we had wind data, temperature data from the cameras, and time of day, all these different things. Like, how is that explaining changes in butterflies? And then we wanted to give the butterflies more space or room to make decisions. And so to like pick a non-biased way, we would, for a given day, we'd find when was the maximum count for that day. We'd start it there and allow that to go to the next day's sunset. And that gave us a longer time frame. We did linear regressions. So we asked simply, does wind speed predict changes in butterflies? And then we did more advanced statistics, what we're calling microhabitat modeling, where we use GANs to control for all of the different variables we had available to us the best way that we could. Built a bunch of different competing hypotheses and compared those models via AIC to see which best fit. Okay, so for the linear regression, here are the results for our 30-minute response time. So again, what it's saying is from, let's say it's 2:30 now to 3 p.m. How are those butterflies changing over that period of time, given the maximum wind speed observed during that time, which we found to be all wind measurements were like highly correlated with each other. And so this we thought was the most biologically interpretable and closest to the original hypothesis. And as you can see, there's like a ton of spread across. So there was large negative changes, but also large positive changes, mostly no change. And this blue line that's hard to see, that's the linear regression, basically saying we can't predict any change from butterflies. The R squared value is effectively 0. And I think what is also telling here is that 2 meter per second threshold, the majority or many of the observations are beyond that. Some going up to 12.5 meters per second. So this is roughly like 6 to 7 times increase in what we would expect, and still showing no change. And when we do the same for the next day response, we have much fewer observations because this is like a day-to-day analysis, but even still the pattern repeats. I know like this line looks like it's going up, but these shaded areas, that's the confidence interval. And if we look at the R squared value, it's quite low. And so this line is not very predictive. We can see, again, spread across either side of 0. Sometimes the number went up a lot. Sometimes the number went down a lot. A lot of it spread around 0. But I think most telling is that for every day that we observed butterflies from one day to the next, the maximum wind speed was always at least 2 meters per second or greater. There was nothing below that predicted threshold. Suggesting that the butterflies at Spring Canyon are not responding in the way that we would have thought historically. Okay, this is a lot of text. I'll just go through it pretty quickly. Basically, we used our maximum speed, temperature information, and our direct sunlight information. So how many butterflies were experiencing light, controlling for the previous amount of butterflies, to more statistically robustly like see, is there a story here? And when we look at if there's anyone who wants to grill me about this later, I'm very happy to go into it, but most people don't want to. So for the next day response, this was like very much our best result from the study. So what we have here is it's a fairly kind of complicated graph, but it's like an interaction. And so it's saying in areas that it's blue, that means there's a strong decrease in butterflies, or like deeper blue. That means butterflies are going down in the cluster size, and red means they're increasing in size from one day to the next. And it's not dependent on just wind or just light, it's the interaction of the two. So if we look at the graph and we hold all butterflies in shade, so we only look along the bottom of the x-axis here, we can see that it's fairly neutral. It's a very light color, which is showing that even as we increase wind speeds, changes in butterflies are not really being impacted. But as soon as we add a little bit of sunlight into this mix, the number of butterflies in direct sunlight, we have this like unusual ridge or band where monarch butterflies seem to be going to those places. They're coming from somewhere else and they're arriving at this location when there's a combination of strong to moderate winds and direct sunlight. And if we hold wind speeds at calm conditions, so 5 miles per hour or less, and we add a lot of light, those butterflies seem to be leaving those places quite strongly. And so this was a surprising result for us. We didn't realize that the two might be working together to help push or pull how butterflies are changing. We did the same for the 30-minute analysis, and the story is similar, but a little bit more confused. There's a lot more data points, and we can see like maybe there's some support for that drop where above this threshold, there's a decrease, especially when there's direct sunlight involved. But on these borders here, we have red again, suggesting that there is some interplay happening here. And I do want to draw your attention to this. 30-minute response has a relatively low R-squared value, while our next day response explained about 40% of the variance. But when we look at other response variables in the 30-minute time window, we can see some subtle but interesting effects. So even controlling after wind, even controlling after light, all these other variables, we do see this consistent pattern of monarch butterflies departing in the morning. So this is essentially minutes since the first counts, minutes in sunrise. And so in the morning, we have butterflies decreasing in size. Around noon, there's an inflection point. And in the afternoon, they're reforming their clusters before settling at the night. So this was a really fun result, confirming what a lot of people anecdotally know about monarch butterflies. And seems to be a temperature effect as well, where there's really not very much happening at these relatively cold temperatures. So this blue band here refers to our flight threshold temperature. This is where we think butterflies need to be at least this warm before they can initiate flight. And so it makes sense. There's not a lot of movement, and there seems to be like an increasing pattern of butterflies potentially at a certain range. And then once it gets too warm, they drop off quite rapidly. Okay. And because I took photos, I could actually show you some time-lapse imagery. So all the butterflies are clustered in this area. This figure here is showing the wind. This is that threshold. And so here's the butterfly. Light comes through, and the butterflies are basically gone. So this is why we started tracking light in the first place, is we would see patterns like this, where the morning light seemed to erase the clusters, even when wind speeds were quite low. Like this, none of the wind conditions, it's like perfectly calm, barely, not even getting to like 3 miles per hour. And those clusters are taking off. Contrast that, here's a high wind, low sun situation. So there's a lot of butterflies, probably several thousand or low thousands in this branch right here. And they are not experiencing direct sunlight, even though behind them they are. Well, wait, and if we look at this graph here, when night comes, here in a second, we have very strong winds. There we go, like several times the threshold that was predicted before, and yet they persist through the next day. And then here's our highest wind. The butterflies are down here, a relatively small cluster, and these poor guys are getting absolutely ragdolled in this like 25 to 30 mile per hour wind. A lot are gone, but later in the morning, they will reform right there. Yeah, you can see them reforming. So they're clearly not deterred by these strong winds. And we can see that in the time lapse, and the results also suggest strongly that this 5 mile per hour threshold is not as strict as we once thought. So if it's not strictly this wind, what might it be? Well, we don't know at this point. I'm suggesting here maybe some alternative explanations, some other places we could look. One idea that came to mind is maybe this is really all just thermoregulation. So monarch butterflies are these ectotherms. They're very sensitive to their body temperature, like I mentioned, during the overwintering phase, because that determines how quickly they use their energy reserves, their lipids. And so they need to find this particular balance. They have to use their behavior to maintain that balance. And perhaps wind is just another tool to help them do that. So they can use sunlight to help raise their temperature. Maybe they can use wind as a form of convection cooling. And finding that sunlight wind balance together is a way to find that Goldilocks zones for them. This thermal image here is not of neither of these are monarchs, of course, but it does highlight that there are pretty advanced structures within butterfly wings across a wide taxonomic range that show these systems for moving heat around. And so I think there is compelling reason to think that they are responding to this quite strongly. And I also think that light perhaps is maybe the better abiotic condition that we should be thinking about. So there's a mystery, like how do monarch butterflies that are separated by 4 or 5 generations routinely find themselves at the same overwintering sites, but not just that, the same trees and often the same branches, even though they've never been there before. It was their great grandparents that were there before them. How do they do that? Temperature, wind, these are all stochastic, random things that happen in the environment. It's hard for us with all our cleverness and our computers to predict it into the future reliably. Can't imagine a monarch's doing it much better. But we do know that their vision is really well adapted. They rely on light and the location of the sun for their migration. And the structure of a canopy of a eucalyptus grove is not changing dramatically from year to year. The pattern of light that those trees create are going to be quite stable year to year, even decade to decade. And we know that when a large, important tree is lost in a grove, we can see dramatic changes in butterfly behavior. And this figure here is one I adapted from Stu Weiss's 1991 paper that was important in establishing hemispherical photography as a tool for monarch butterflies. And what he showed, like going to a bunch of these different groves, is that permanent sites, those that represent the highest suitability for overwintering butterflies, those that maintain monarchs throughout the season, they have a very narrow range of percent open sky. This is the total amount of open sky from his photos. As we go to an okay but not great site, a transient site for monarchs, that range increases significantly. And then for unoccupied or former sites, we have this massive range going from very dense, almost completely closed, to almost entirely or like halfway open. And so this is what I'm hoping to look at next. I think there's definitely something to be explored here, and it could mean quite important things for management of these overwintering groves if light is shown to be a critical abiotic factor. So in conclusion, our study does not support this 5 mile per hour, 2 meter per second wind threshold widely used in conservation plans. We think thermoregulation is a better way to think about how monarch butterflies are selecting and utilizing these groves during the overwintering stage. We think light is a great place to look next in terms of understanding monarch butterflies, and partaking and understanding these science-based management practices are really important for monarchs, particularly in the West. Like we're in our new normal is like low 10,000, and that's like terrible for these butterflies. And so we need to know everything we can so that we can help them. So with that, I have a lot of folks to acknowledge. This study was made possible by a lot of really wonderful people and a lot of organizations, in particular Dan Meade, who was my mentor at Althouse Meade, who got me started on this whole thing. So with that, I'll take any questions.
Wow, that was really interesting, Kyle. Thank you so much for that. I love the time-lapse pictures that you had. That was cool to see. Are there any questions for Kyle at the moment? Well, I had one for you.
Okay.
And I know you kind of were seeing it in a grove with bigger clusters. Do you think that your conclusion holds true for groves that have small clusters or like individuals?
Yeah, it's hard to know. So my thinking on this is, and I'm in like a bit of speculative territory here, but because the monarch population has been low for so long, I suspect there's a lot of potentially suitable habitat out there already. And because monarchs clearly don't randomly arrange themselves, they will congregate very strongly, aggregate together. So maybe there's like other places that they could be, but we just happen to find them at this particular location during this year.
Mhm.
Like one way I think about it is like, okay, you have a hotel, there's 100 rooms, but there's only like 5 butterflies to occupy those rooms. So they're probably always going to take the top 10 rooms year to year. And there's still like 90 other rooms that are good, but we just don't see them using it. And we won't know the full range of that habitat until the population recovers to like 1980 levels. So in terms of these um, small groves, like that may just be an artifact of these very low population sizes. And it's hard to know the true range of monarch behavior until they can fully express themselves.
Interesting. Cool. Thank you.
Yeah,
Um,
thanks.
okay, cool. If anyone has any other questions for Kyle, definitely drop them in the chat
Um,
or reach out to him.
Let me just address like Deborah's question
Yeah.
really quick. So yes, grove composition and structure continue to be like they're pointing to be very important for monarch butterflies. A paper came out recently by Peter Ibsen and other authors who was involved in this research as well, who looked at Pismo monarch grove from a different bunch of different angles, and the primary result there was the the structure was like critical for understanding why monarchs are where they were, and like particular trees were especially important as well. So there's something going on in the configuration of that like large woody area that these butterflies seem to like. And it is also helpful. I'm sorry, I'm eating into a lot of time, but it's helpful to think that like monarchs' exposure to eucalyptus is like no more than 120 something years old. So they're quite adaptable species. This is not like a deep evolutionary relationship. It's them being opportunistic. So I think there's a lot of hope and opportunity that we can take care of them going in the future. Okay.
Awesome. Thank you so much, Kyle.
Thank you.
And with that, I will open the floor to Charis, our next presenter. If you want to share your screen, we can get going on your cool project that you're working on.
Okay. Okay. You see that?
Yes.
Okay. Let's see. Great. Okay. So thanks, everyone. My name is Charis Vanderheide. I'm a senior biologist, monarch butterfly specialist with Althouse and Meade. And I'm here to tell you about how we are using radio tags to understand monarch overwintering behavior. So let me see if I can skip. Okay, so I will very briefly talk about overwintering habitat because Kyle did a fantastic job of describing that. I'll talk about the history of monarch tagging, the history briefly of wildlife tracking technology, and then our tagging study. Oh, first to go back, this was an incredibly collaborative project, and I'm so grateful for everyone that was involved. And I have hopefully everyone's logos here. Okay, and I'm going to skip this because we went over what makes good overwintering habitat. And okay, so very quickly, the Western monarch count, we all know, is a fantastic way for us to count monarchs, understand the how the population is doing in the West, and understand how monarch butterflies are using each individual habitat and where they are in that habitat. And here's the graph. This last year was devastatingly low, like last year, and I'll quickly go through that. Okay, so, and counts are static. It's one shot. You visit once, you get one count. Monarchs move, and we have so many questions. So some of our questions are, how do monarchs use overwintering habitat on the individual level? What does overwintering habitat behavior look like from the day-to-day? Are there behavioral patterns? How often do monarchs venture out to find nectar and water? How far will monarchs travel and then return to the site? How far will they travel and then not return to the site? When do they leave the site? Where do they go? And we can kind of infer some of these questions from the monarch counts. Like if we're looking at a site that has a lot and then none, and we're looking at another site that has none and then a lot, we can infer that they moved, but we don't know, and they could have gone somewhere else. Or anyway, so we have so many questions of movement that would help us understand how they're using the sites. So monarch tagging started several decades ago. When I first got started researching monarch butterflies at Cal Poly about 26 years ago, we used a fine tip Sharpie marker to write a 4-digit number in the cells of the hind wing of the butterfly, right here where my cursor is. And then people developed stickers with a identifier number and a 1-800 number or an email. And that's dependent on someone finding that tag. You know where you put the tag on, but then you need someone on the other end to call you up and say, I found your butterfly, which was amazing. And we got incredible data from that. And then you have the end and the start and the end point. And then at the same time as that was happening in the monarch world, we had the MODIS tracking network that was being developed across the Americas, and they were tracking mostly birds. And so these radio tracking tags were being applied to birds, and then this image shows how birds have been traveling across North and South America for many years. And so us in the monarch world have been slowly watching these radio tracking tags get smaller and smaller and smaller, just waiting for the time when the technology finally gets small enough that we can put it on a monarch. And last year, about 2 years ago, it finally happened, and we were so excited. So the cellular tracking technology, CTT, came out with the blue morpho tag that is now small enough to put on a monarch butterfly. And so this tag sends out a detection on the 2.4 gigahertz frequency, also through Bluetooth and it can be detected on Bluetooth enabled devices. It has a solar panel on it and no battery, which is how they were able to keep it so small. So this tag weighs 0.06 grams, sometimes 0.07 grams. A monarch butterfly's weight is about half a gram, so 0.5 grams. And so that is about 12% of the monarch butterfly's weight. So in our terms, that's about a 20-pound backpack. So when you go out for a hike and you're wearing a 20-pound backpack, you're going to feel it, but hopefully you're still able to make it up the mountain. And the same thing that we're seeing with the butterflies, they're still able to fly from Canada down to Mexico carrying this tag. And so this tag is powered by a solar panel and will only send out a detection when it is receiving sunlight. Okay, so in anticipation for this study, we set up a MODIS tower at Elwood Mesa, and this was with the help of the city of Goleta. And in order to do a monarch tagging study at Elwood, and we can pick up the detections of the monarchs at this tower. And so we developed the Central Coast Monarch Study, and our goals were, one, to develop or to validate the tagging methodology. Because with new tags, we were developing, how do we do this? How do we put it on a monarch in a healthy way? And there were fantastic researchers that were doing some incredible research to look at what glues are best and what is the process that you can put a tag on a butterfly, have it stick, and also increase the longevity of the monarch. And we also wanted to look at the behavior of these butterflies with tags. Can we better understand overwintering monarch behavior on an individual level and better understand habitat use, resident time, site fidelity, movements among sites, movement rates, the home range, meaning how far do they go on a daily basis? Is it small? Is it large? What is the average distance that they travel for nectar or resources? And then survival. Okay, so really important thing to think about when you're designing a study is your study design. And it starts with a question. And then you need to figure out, how do you best design your study that the data you collect will help you answer your question? So a good study design is as symmetrical as possible, as standardized as possible. A large sample size is best. So we figured out 10 tags per site is a good number. Also, we had low population year to consider also. And then sites are at a fixed interval apart. And then you have to take in field constraints and budget and staffing constraints and feasibility. And but most importantly, will your data answer your study question? Okay, so biologists love a bell curve, and this is just fantastic. So this was data that we collected from Elwood, Maine in Elwood Mesa, back when there was a significant amount of monarchs present. So this was 2010, 2011, and you can see the monarchs show up in October 1st, and they leave March 18th, and they peak just before Christmas. And so we decided that we were going to tag here mid-November and hopefully still be able to see behavior through March. So we had our time window. Okay, so we designed our study to tag 10 monarchs at Elwood Mesa, 10 monarchs at Gaviota State Park, and 10 monarchs at Pismo Beach. We tried to stick with equal numbers of male and female, and we got 16 females and 14 males. Okay, so Kyle talked about overwintering habitat. Our first goal was to go out at these overwintering sites and find the monarchs. And this year was quite challenging because there weren't a lot to choose from. So, but when we found them, we used a net on the end of a very long pole. This was a 28-foot pole, and we reached up into the trees with the net and surrounded the cluster with the net, and then gently shake. And we went really early in the morning because Kyle was talking about that flight threshold. It's about 55 degrees. So when it's colder than that, they aren't able to move their wings and fly. So when you agitate them just enough, they will fall into the net and they will not be able to fly out, which is convenient. So then we would lower the net down, collect them, put them in this basket, and they would crawl up the sides and keep roosting with their wings closed because it was still cold. So then we applied the tags, and the method that was devised by Lee Brown and her researchers, Dr. Lee Brown, was to use eyelash glue. So it's non-toxic. We tried a bunch of different glues, but eyelash glue was the best. It dries quick, it's non-toxic, and you paint a little bit on this tag, right here is my finger with the tag. It is so tiny. And you paint a little bit on the monarch and then you press it on very gently. And this looks extreme, but what we're doing is we have a glassine envelope over the monarch's wings, so it's not going to get damaged. And then we used a wide washer that could distribute weight evenly across the wing, so it wouldn't crush the veins in the wing, and hold it steady so that we can apply the glue, and it stays there maybe 5, maybe 10 minutes if it's colder and the glue is taking a little bit longer to dry. And then we take some measurements, and then we would put it back in the net, butterflies back in the net to hang out, acclimate back to being a butterfly that is not penned down by something. And I have a little video that shows these happy butterflies ready to be released. And can you hear that? Can you hear? So I just love that video because you can hear the flapping of their wings. And if you ever get a chance to go down to Mexico, you can hear millions of those wings, and it gets loud and it's phenomenal. So anyway, little travel plug. Okay. Oh, there we go. Okay. So then we would release them, and we would release them into the trees. And this right here was our first butterfly that we released. We named him Danny, and immediately he flew out of the net and directly onto this branch, wings open in the sun, tag is there, getting detections, and he just behaved beautifully. It was fantastic. Okay, so then how are we getting the data? The data is coming a few different ways. One, it's getting detected by our MODIS tower out at Elwood that we installed with the 2.4 gigahertz receivers. We also installed a network of nodes, and we put this is a way to expand the network. And so if we get a detection from the node, we have better locating data to know exactly where that butterfly was. And then the data is coming through the Project Monarch app, which I hope most of you have already heard about, and also Bluetooth enabled devices. So here's the app. If you haven't heard of it, you could download it on your phone. You go out to the grove, you hit detect and start scan, which is right here. And then on your phone, bing, 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 all of these, if there's butterflies in the area, they can pop up and you can see the name and the signal strength of how close they are to you. And this is a way for you to know if your phone actually is helping detect butterflies. And without this app, your phone still could be being used to help locate butterflies. It works more like an air tag, but this is the way for you to actually know that you're contributing. Okay, so this data, we could see our tagged butterflies in real time, especially with the app. And so Micah, in the first 3 days, he was tagged here in Pismo Beach, and he flew all around Arroyo Grande, Grover Beach, Oceano, visiting all these sites. And then at night, he would come back to Pismo Beach or Oceano, kind of almost interchangeably. And then you can see here all of our butterflies. And then these were all the butterflies that were being tagged across North America. And since I took this screenshot, butterflies have been deployed in Mexico as well, and getting some really incredible data. Okay, so our longest distance flight was a butterfly from Gaviota State Beach, and it flew up to Pismo. And that happened, I think in December, so pretty quickly. We also had some butterflies from Gaviota explore westward. None made it. I mean, this was about the farthest that they made it. They didn't go all the way to Elwood. And then this one was really interesting. Bruce, he flew around Grover Beach and Rio Grande, and then, excuse me, he spent a long time in this spot, which is a new overwintering site, or an overwintering site that we hadn't heard of before. So he's helping us potentially find new suitable habitat. Okay, this was fascinating to me. So I was really curious about home ranges and how, like, what is the size of the area that an individual overwintering monarch is using? And based on these three individuals, it says it really varies. This butterfly was very close most of the overwintering season. It was about 0.1 miles across, or about 200 meters. Angela was about 320 meters of a circle of habitat. And then Mia was over 1,000 meters across, 0.7 miles of habitat that she actively visited over the overwintering period. So not a straightforward answer, pretty much butterfly dependent or individually dependent. Okay, then I was also really interested in tag retention. Was there a time where, like, how were the monarchs dealing with having a tag on? And I'm encouraged by this graph because this shows on day 0, there were just shy of 30. I think one of our tags fell off almost immediately. So, but 29 did really well. And so this shows that there is a pretty much evenly gradual decline of butterflies going offline. So either they die for some reason, or they get buried, or their tag falls off. I don't think that's the case, but it shows that there wasn't a set time where the butterflies would all of a sudden stop being able to fly. So that was good to know energetically. Okay, this is another way that I portrayed the data. So this was, each of these orange lines is the start and end date across the season of when the tag was actively being detected. And so that also varies. And I was really interested about these last 3. So we jumped around, and these last 3 we tagged at Elwood Mesa, and they were had a great survival rate. And then I was curious about storms. So I looked, and right about here, Galita had a big storm. Another storm came through here. Another storm came through here. So I was interested, can we see if monarchs are becoming undetected after a storm? And for this quick and preliminary analysis, I would say no, they survived Elwood pretty well after storms. And then interesting point here, Orion has almost the longest detection rate. However, the last month or 2 of his detections are about in a pattern that we think that the tag is still active, but the butterfly is not. So it's about 3 detections a day and the same signal strength. So that means he's probably in the exact same spot, and the tag is just getting a little bit of sunlight and detects when it finally gets enough. All of this to say, we have millions of data points, and it's going to take an effort to get through it all and really tease out what the data is really saying. So these are preliminary. Okay. How am I doing on time? Almost. Okay, I'll be quick. Okay, so spring dispersal was another thing that we were really hopeful that we would be able to collect, but we weren't sure because we didn't know how long a butterfly with a tag would survive. And surprisingly enough, at least 6 made it through to March to show us where they went after overwintering sites. So here you can see a really brief snapshot of the 6 butterflies that left Elwood Mesa. So here's one, Dennis. He left Elwood Mesa and flew around Santa Barbara. Norma Jean hung out at Elwood Mesa and left Elwood Mesa and spent some time at Los Caneros Lake, which has 3 overwintering sites that we know of. And then she spent some time in private property, and it doesn't look like overwintering habitat, but it could be, or it could be breeding habitat. I'd love to know. And so that was interesting to me that what I would think is spring dispersal, she was spending time at another overwintering site. And then what we really hope to learn is, can we use this habitat behavior data to inform habitat restoration and conservation? Can we understand where we should be planting nectar plants? Is there an opportunistic or a place that would be better suit the monarch butterflies? And how much of the overwintering site should we be protecting and conserving? How much do they actually use? So there's some really interesting things we hope to learn as we dive in. And then we all know this. Hopefully the thing we can do in our gardens is plant winter flowering nectar species, plant native milkweed. If you have the tropical milkweed, cut it back in about December so that you can have fresh new leaves in the spring. And pesticide, herbicide-free gardens is best. And then just a shout out to everyone that helped. We had a great time and a great team. It was a super collaborative project. And we just had so many people coming together to make this possible. The Xerces Society helped us get a scientific collection permit with the California Department of Fish and Wildlife. Elwood Friends contributed some tags. The U.S. Fish and Wildlife Service Postal Program provided a grant that made all of this possible. The city of Goleta helped fund the Modus Tower and the maintenance of the node system. State Parks was so amazing at getting a multi-district scientific collection permit for us to go and pivot when there weren't many monarchs at Elwood that we needed to. We could pivot to Gaviota and Pismo remarkably quickly to capture that range of the overwintering season that we wanted. And then Althouse and Meade, which is where I work. And then all of our volunteers were incredible. So with that, I can take some questions.
Thank you so much, Charis. That is so cool. I love it. I think we have time for one question before we move on. Does anyone want to speak up, or I can just read one of them from the chat? Are you doing this next year too? Or this winter?
I hope so. Funding permitting, but yes, I hope so. And I would love to expand into Ventura to tag 10 butterflies in Ventura, and then Elwood and Pismo or Gaviota. So cross your fingers, yes.
Cool.
Yes.
Can you use the tags if you find them?
Oh, yes, that's another thing. I absolutely would reuse a tag. And if anyone has the time and desire to go out and hunt for these tags, please don't take one off a living butterfly. But if you find them in the wild, yes, please contact me. That would be incredible. And if we want to work on a task force to go out and mobilize some searching efforts, that would be incredible. Yes. Because the tags aren't cheap.
Yeah. Okay, cool. Thanks.
Yeah.
I think there was another question in the chat for you, but I'll let you answer that that way, and we'll move on to our last speaker before we kind of get into some overwintering protection updates. So I'll have you stop sharing your screen, and we'll let Alaric share his.
I don't need to share a screen right now. I would rather present just face to face, as it were.
Sounds good. Do what you got to do.
Okay, we're ready?
Yeah.
Yes, my name is Alaric Balibrera. I run a nonprofit up in Santa Rosa, Sebastopol, Central Sonoma area called Rainforest Rising. And what we do is we enhance seeds to enable direct seeding. Our mission is to scale up restoration and to accelerate restoration. And we understand that it's very difficult to scale the planting of plugs. It's very labor intensive, costly, time consuming, etc. So what we set out to do about 7, 8 years ago was figure out a way to enhance seeds so that they can compete with invasives, and so they have a healthier set of growth metrics. So in other words, their germination is accelerated, their growth is accelerated. We are also about to delve into a fungal inoculation program based on a study that came out of the University of Kansas a couple of years ago, showing that their fungal inoculant in their milkweeds in Kansas doubled the size of their milkweed. So this was very exciting for us, and we're about to hire on a mycologist and start a fungal inoculant program for our seed balls, which is another enhancement that we are developing, so we can fungally inoculate the seeds and accelerate their germination, and then put them in a seed ball and drop them from drones. That is basically the idea that we've been working on, and it is going great right now. Let me see. I can probably share my screen at this point for a couple of, let's see. So, can everybody see that screen?
Yes,
Okay,
you're
great.
in presenter mode or like, yeah, but we can't see the screen.
You can't see the screen. Okay, that's fine. That's great. What we have is we have been funded by the state, by the Wildlife Conservation Board, to start creating or help add additions to the Monarch corridor through Central Sonoma, which is a sort of a desert for monarchs around here because the human activity has well, between the humans and the invasive grasses, the flowers have nearly disappeared. So we are going with these sites. If you see here, there's Santa Rosa, Sebastopol, more Sebastopol. Down near, we've got a site at Sonoma State University, and there is a very large site that's being restored in like a very urban area of Santa Rosa called Colgan Creek. And there's a great deal of activity, a lot of momentum around the restoration here of a monarch corridor. But I wanted to get in a little bit about what we're actually trying to do. So here is a picture of our some of our seed balls, and there is a seed ball cut in half with a wheat seed inside of it because you have to do a ton of trials. Here is some inoculant that we pulled out of the rhizosphere of a healthy milkweed stand and then reproduced. We cultured and reproduced in the laboratory, and a drone which we are also developing for large terrain monitoring with these. So you'll have to excuse me because I'm all over the place, but that's because what we're doing is all over the place and everything is interconnected and interdependent. So I jump from subject to subject, but it really is all connected. Um, the monitoring is a thing that's coming in hot with AI and with the infrared and the near infrared and the other multispectral bands that can be captured on camera now. You can see, for example, where there is a lot of moisture, where there is dryness. You can see the soil profile, so you can find out the soil pH. You can even find out the nutrient profile of the soil. Sometimes you can take a wide array of health metrics from the vegetation, and we are learning how to identify vegetation. That's coming along pretty slow. It's difficult because you have to upload 10,000 pictures of an oak tree in order for the AI to begin recognizing oak canopy. And so, as opposed to whatever bay canopy or whatever they're looking at. So that's what we're doing, and you can see here the little sproutlings emerging from the seed ball. And so, what we are hoping to do with a number of key pollinator species, including Fascicularis speciosa, cordifolia, a couple of grasses, Stipropulchra, and Festuca rubra, as well as a few late season bloomers, because here we're close to the coast and we want to provide nectar for the monarchs as they come back in and as they depart. So we need the flowers that are blooming in February and March, and then again in October and November. Um, so that's what we're planting at our habitat sites is kind of like extended season bloom for nectar availability for the monarchs as they pass through early and late in the season. And and so we are designing these seed balls here in order to have, say, half a dozen species in them. So if you throw them out and they are fungally inoculated, fungally enhanced, and they are enhanced. I'll explain what enhancement, like the seed enhancement technology that we're using in a second. But you can actually compete with invasives, and they have a greater threshold of germination in temperature and a greater threshold in moisture. So that, in other words, they can germinate with less moisture and they can germinate in both higher and lower temperatures. So those windows are expanded, and so you get kind of enhanced competitiveness in your seeds. So what the enhancements are is that they are not genetic and they are not chemical, they're not any kind of polymer. We are simply like optimizing the natural metabolic processes of the seeds themselves. So the primary enhancement we use is called solid matrix priming. And solid matrix priming is, let me give you a nutshell of what a germination profile looks like for every seed in the world. First, the seed drinks, that's called phase one. Second, the seed once it gets enough to drink, where there's enough moisture in the seed, usually that's like 40 or 50% of the seed's total weight. Once it has enough to drink, it starts an interior metabolic process. And it goes through that process depending on what seed it is, the individual seed and the species that it is, takes a certain amount of time, can be anywhere from 2 hours to 40 days or longer, depending on moisture content, temperature, etc. And so when the radical emerges at the end of this interior metabolic process, that process is called phase two. And when the radical emerges at the end of phase two, you've entered phase three. So the trick is to be able to get all the seeds uniformly to the cusp of germination and then stop the process. You can, at that point, you can withdraw them from the process and safely store them. And then all you have to do is wait for a favorable weather window. Like you can wait for 8 months for a favorable weather window. And but if the radical emerges, then the seed either has to germinate fully or die. And so it's very tricky because you're trying to measure an invisible process. And the way we do this is by doing it over and over and over again with the same species and finding out how long it takes them to reach phase three, how long until radical emergence. So the solid matrix priming that I was mentioning we use is a process of putting the seeds into a container with sterilized sand and vermiculite and a very, very small amount of water, which for whatever reason allows all the seeds to drink at the same rate because one of those of you who have tried direct seeding know that the seeds vary tremendously in when they germinate. The seeds from the same species and the same lot even vary tremendously in how long it takes them to germinate under identical conditions. So by doing this extreme regulation of the water that we give to them, somehow all of them drink the same amount for a specified period of time at a specified temperature. And we do enough trials to know, okay, if we give them X amount of water over 28 days at 20 degrees Celsius, we will get all those seeds to the threshold of germination. And then when we pull them out, we store them, we wait for the favorable weather window. And when we know it's going to rain, we disperse them onto the ground. They have to have soil contact with the soil. And they should germinate if the temperature is high enough. If it's within their temperature threshold, they should germinate right away. And so give it our intention is to, for example, narrow leaf milkweed takes anywhere from 4 to 16 days to germinate. And our goal is to get that window down to 4 to 16 hours so that it, and the reason this is so important is because, again, those of you who have tried direct seeding, you know there are some serious challenges to direct seeding, and that's why nobody does it anymore, is because most of the seeds, if you're doing grass, for example, or flowers or anything that an ant can carry off, the ant will carry it off, or the birds will take it, or the whoever will come, because everybody loves a good seed for lunch. And so I know that our collaborators have lost upwards of 95% of the grass seed they put out. So they don't do that anymore. It's not at all cost effective. So so what we do is we pellet them, we pelletize them. We've done many hundreds of trials trying to get the play right between oxygen flow inside the materials and retention of moisture inside the seed ball. So the seed needs both air and water to be able to germinate. So we played with that. We believe we've come up with something that's pretty good. It's not like it can't get better, but it's pretty good. But what that does is it provides the seeds in that seed ball a housing that protects them from predation, mostly. In a smaller way, it protects them from desiccation and exposure and pathogens. But essentially, most of the problem with direct seeding is it just gets eaten. And and so that's one way to protect them from predation. And then they've got an accelerated germination. So this, especially where we are in Sonoma, we are wholly overrun by invasive grasses, and they so what defines them as weeds for us is that they have a shallow root system. They set seed very quickly. They have an enormous amount of seeds, and they grow fast. And they also set out this kind of like this root network that becomes like a matting, and nothing else can grow. There are very few species we know that we can grow. Although I will say, um, just by coincidence, we found out if you are doing any of this and you're facing intense invasive pressure, Phacelia tenicetifolia, lacy Phacelia, is is can compete with invasive grasses to an extent where we haven't found any other species that could. And in addition, as an added bonus, but wait, there's more. They're more or less deer resistant because the deer don't like that fuzzy stuff in their muzzle, I guess. Um, so in any case, that's what we're doing. You can see here we've got the seeds coming out of the seed pod. We can fabricate seeds by the many tens of thousands, or we are developing the cat fabrication of seeds by the tens of thousands. Um, and here is some of the inoculant that we've been using. Here is a picture of one of our sites at Sonoma State University last year. All this purple that you can see here, that is all Lacey Phacelia and obviously poppies there. Um, Here is what we call an eco ball, also just the probably 6 or 10 species in this eco ball coming out. Um, and as I want to show you this study, it's one of many, many that we do, but it's an example of the enhancements that we're trialing. So this blue line here is the control, and the 5 is in days. This is 10 days here. So all of the enhancements that we trialed got the seeds germinating in one day. They started germinating as opposed to starting here at 3 days. By 3 days, all of these are up at 60 to 90% germination. So it really makes quite a difference if you can get this. This is a methodology that is in common universal usage in agriculture. And the that we know basically everything there is to know about the germination profile of a tomato or a soy plant or corn or what have you. And that's because they have teams of researchers working since the 50s to figure out how to make these seeds grow larger and healthier and increase their yield as a crop. And so we were able to read all the stuff. They keep all this information proprietary. They won't give it out. So we went back and we read, most of the studies that had been done since the 50s, and we were able to extrapolate the formula and calculations needed to be able to kind of establish this methodology, this solid matrix priming methodology. Here's another example, a little probably easier to see. This is a Aleppo pine in the Mediterranean. Our sister lab, our research director and sister lab are in Spain. So a lot of our studies are done on Spanish species in the Mediterranean. In fact, this last year, unbelievably to me, we got a 100% establishment rate in a season in Andalusia that didn't rain one time. And so it's very exciting to me because it seems to have a lot of potential to really be able to restore at scale a lot of places that are currently unreachable, like out of our ability to do this. So this this Pinus halepensis, here you can see the first germination in the control at 11 days. And the first germination in a pre-germinated um, protocol started at 1 day. And we've got this green one, which is a another kind of enhancement that we do, starts at 3 days and is at 80% in 10 days. Whereas the control hasn't even germinated one. One of them hasn't germinated at 10 days. So it's a very exciting methodology for me. I really hope that it, excuse me, can capacitate restoration of pollinators at scale, because for certain up here in Northern California, the monarchs are doing terribly because there's nothing to eat. Um, and if we can get the nectar flowers and milkweed, which has been at the top of the list of extermination, um, we can potentially save them from extinction. So I'm very excited about this. I wanted to present this because we really want to help anybody who wants to create pollinator habitat anywhere along the coast or inland. I will put my information, my contact information in the chat, and you can reach out, and we would love to be able to help you with wherever you are. So with that, I think I should probably take some questions. There may be some. Um,
That is
uh,
pretty cool. Cool research, and I'm glad to see that you're having some good success with your seed bombs. You said you've been dropping them more in other places, some of the sites you're looking at now.
yes, we some of the sites that we are actually building, we're doing field trials with the seed balls.
Cool.
Yeah,
That's
yeah.
incredible.
We have not, however, yet completed the development of the priming protocol, of the solid metric priming protocols for those species I was mentioning. We will have probably our first protocol available in later this month or May. And we would love to pass those protocols out also. We are not just nonprofit, but also open source. And we believe this should belong to humanity. So we would love to pass the priming protocols along to you as well, so you can prime your own key species. You can do the research that's required to find out, like, what are your key species in the area that you live, and what do the monarchs and the native pollinators need most. And if we can work together to develop that priming protocol for that species, then we'd love to be able to do that. And if the key species you need, we already have a priming protocol for, then we can work together to get your seeds primed.
That is awesome. Yeah, we'd love, I'm sure, down in Southern California to include this in some restoration activity down here. And it seems like there's a couple people in the chat who agree and want to talk more. We do have a couple questions. One is, what are your oldest sites you've been working on?
The oldest site, in what sense? In the sense how long we've been working on it?
Yeah, I guess
Uh,
so.
3.5 years.
Okay. And do these seed bombs or your protocol work in dry habitats?
They work everywhere.
Awesome.
They work in all systems because it's simply accelerating the germination of the seed.
Okay,
I
cool.
mean, if you put them down in a weed patch, your weeds will, right? So, I mean, it's, and in that case, I'm talking about the kind of like the inoculant, like biologically enhanced, liquid biological enhancements with lots of bacteria and protozoa and nematodes and all that stuff. If you put that onto your weeds, the weeds will love it. So you have to be discriminating, with what you're doing, but But the enhancements themselves should increase both like biomass and growth health metrics, no matter where you're planting them.
That is awesome. Awesome to hear. Okay, cool. Does anyone have any more questions? If not, you can put them in the chat. Alaric has put his contact information there. You can reach out, discuss more about seed bombs and including that in your restoration. And if not, we will move on to our last little bit of business, I guess you could say. And I'll hand the floor over to Chris. Thank you so much to all of our wonderful presenters on their projects and research. It's been incredible. So yeah.
Okay. Rachel, can you hear me?
Yes. we can
Okay,
hear.
perfect. Okay, let me see if I can start the video here. Wow, great presentations by everybody. I'm stoked about the radio tags, about these seed bombs, all sorts of stuff. My name is Chris Amendt, and I am the founder of Native Monarchs, which is a Ventura County nonprofit. We are dedicated to creating new habitat with native milkweed, and also timely, because it's happened just recently, is protecting existing habitat like overwintering sites in Ventura County. I guess for some of the updates, if you weren't aware of it, back in December of 2025, the owners of Ventura Coastal LLC, which is located on Vista Del Mar, started cutting down the trees of a designated monarch overwintering site that's recognized by Xerces. That had always been on the radar for a long time because Ventura Coastal was looking to sell that property to a developer, and then the developer wanted to come in and put about 200 plus homes on there, and this was an ongoing thing for years with Ventura City. So I was always kind of worried about that in the background, but it was the week before Christmas. Code enforcement wasn't working. There was nobody around. It was a weekend, and they started cutting down the trees. Located in the coastal zone without a permit. So that kind of like opens up the doors to property owners that are located within the coastal zone that think if they want to sell their property to a developer, that the developer will often ask, you need to remove these trees if they're going to be a problem in the future for our development before they do the deal, before they transfer it over. So that was the first problem. We were able to get a stop work order on this. A whole group of people showed up, media showed up, police showed up. But basically there was no permit issued to take these trees out. And then there was a stop work order on it. So even if you go by Vista Del Mar now, you can still see the remains of what trees were cut down. They cut down about 8 of the large eucalyptus trees, but basically just left them there. So there's still a lot left. And I will say about this property, the Vista Del Mar property is actually, the overwintering site is located on 2 adjacent properties. So therein lies another problem. If you think about it, if 2 people own the property where these trees are, then you got to get the buy-in of both people as far as that. But as far as what's happening now, I'm going to stick to what's out there on public record, and the project details are on public record as far as the design of what they want to do, as far as the houses they want to put there. It's already in print, and it shows all those trees are gone. 35 trees, including all of the ficus trees at the beginning of that property, are slated to be eliminated. And then there is a pocket park that's put back in the corner, which I think is for, people and their dogs, and it's got a couple of ornamental trees. But all those eucalyptus, the cypress that are still on there, and the redwood, as well as the really mature ficus that are in the beginning of the property, were all slated to be removed. So we are continuing to show up for city council meetings. When I say we, it's a small group of us, but and I know, and I'll just be completely blunt about this. I know that there are a lot of people that work for government agencies, state agencies, Fish and Wildlife, Coastal Commission that are on this call, or you work for the city or the county. I understand it's an awkward situation to be in where you can't basically give your opinion on this because it's kind of it's awkward for you guys. But in the long haul, I work for myself as a nonprofit. So if you can help us, if you can bring something to the table that basically says you shouldn't be doing this because, bring it to me. I have no problem bringing that up. I don't answer to anybody. I'm not afraid of anybody, but I want to really preserve this site. And I think if we do it right, we could actually put a conservation easement on this area, on this trees for these two properties, and make it into a monarch preserve, kind of like they have in Pismo. I know it's a far stretch. There might be something where, we have to raise money and pay for it. And that happens all over right now, is that people are putting funds together to protect. It's the only way you can protect this land, if you buy it. So if you leave it up to chance, my opinion is we're going to lose it. But you can always come to me, offer your support, offer your expertise, because I'll take it. And then I will go back to my group and we will put in the public comments before every city council meeting. We will show up to city council meetings and we will read these letters. If you want to do it anonymously, I'll do it. But I think that is our best bet, because we're looking at public record now, because the ban that Ventura City put into place, the temporary tree cutting ban as of February 10th, with the fines and everything, you need a coastal permit to cut these trees down. But any other tree that has wildlife nesting in it too. So there's a ban on that until the tree maintenance, the mature tree ordinance is in effect. But there are permits coming in to remove these trees, and then that has to be looked at too. If they do it the right way, that's great. But if it's basically for development purposes, who's going to approve it? Do we know? So we're kind of at that. We really don't know what's going to happen when these permits come in. Do they approve it? Do they look at it? Is it a designated overwintering site? Is it ESHA? Is it part of CEQA? Those are all the questions that kind of need to be answered. But so far, no other tree cutting activities are going on at Vista Del Mar. And hopefully we will have some more information in the next month or so about what has been discussed with the city, if they want to share it with the public. And I welcome any recommendations, any questions. But honestly, this is going on up and down the coast of California in every coastal town. Anytime they want to develop something and there's a grove of eucalyptus trees, and probably there's monarchs there in the winter, it's always a fight, and it's going to be on a city level, and also a state of California level, because we know that the federal protections probably are not coming anytime soon. So I'm not waiting on that, but we're working on it on city level, county of Ventura, and also state of California level. So with that, any questions, let me know.
Thank you for that update, Chris, and all your hard work with the city council and pushing to some protections for the overwintering sites and monarchs. Does anyone have anything to add or say regarding that specific topic?
And if anybody wants to me privately, you can email me at Amendt, CAMENDT, at nativemonarchs.org. But I mean, honestly, everybody on this call, this impacts everybody because I know that we are all invested in this monarch butterfly. The Western population is collapsing, and all of these studies and things like that. We need to preserve these sites in order for these studies to go on, and we can find more data to protect them. And I think that's the basis of it. You got to start at the level where they're starting to destroy the habitat. And that's where we're at, is trying to preserve this habitat.
Cool. Thank you so much, Chris.
Thank you.
Okay. Oh, Abby said, I have an update on future changes to the County of Ventura tree protection changes. Abby, did you want to speak up at all? Oh, perfect.
Hi, sure. Chris, great job. I just want to make people aware, over this pretty much the last few months, our Board of Supervisors have been really focused on permit streamlining. And there's going to be a number of changes to our standards. And when for example, a ministerial permit, which is an over-the-counter permit where they just need to meet specific standards, versus a discretionary permit where it's a more involved process. They're subject to CEQA, they need to have a hearing, those types of things. And so one of the proposed changes is that we make all of our county tree permits ministerial, which would mean that no mitigation would be required. Um, People, if they didn't necessarily wanted to speak out about it, they wouldn't have that opportunity. So I can go ahead after this meeting and forward you sort of the last board meeting where they outline sort of those changes ahead. And I just want to make people aware, particularly this group, because trees are so important, sort of about those changes. So thanks.
Thanks for that update. And yeah, if you want to send it either to me or put a link in the chat for the board meeting, I can either distribute that via the MRAC listserv, or people can go and use the link that you have from the chat. So
Yeah,
thank you.
great. It's going to take me a little bit to get the right time, see the whole hearing, but I'll email it to you after.
Okay, sounds good. And I'll distribute that to everyone so they have that update and change that's coming.
Yeah, you guys did such a great job. This was such a good meeting. Thank you.
Oh, thank you so much. We appreciate all the speakers that came today to share their work. So round of applause goes to them, honestly. Okay, cool. Well, does anyone have any last minute things they'd like to talk about, or updates, or projects that they have upcoming that they need support on, things like that? Jaya did just drop our next Monarch and Pollinator planting event in the chat for you all to see or sign up for. It'll be April 17th, 9 to 12 p.m. We're going to be planting a pollinator garden and some trees at La Jolla State Campground, on the border between Ventura and Malibu kind of area. So sign up, come plant with us. It'll be really fun. It's a beautiful spot right on the coast. But other than that, does anyone have anything else? Okay, well, seems like no. So we're gonna end the meeting a little early, and you can go on with the rest of your days. Thank you all for joining us. And we'll see you at the next MRAC meeting, most likely in October or November. So be on the lookout for the next date that'll be. Thanks, everyone. Have a good afternoon.
Thank you so much. Bye.
It's recording.